In this session, we are going to look at the histology of the urinary system. And the urinary system is composed of the kidneys, the ureters, the urinary bladder, and the urethra. But urethra I have not looked at here. Welcome all. My name is Wallace Munyere, and I'm the URE doctor. So the urinary system. The urinary system is composed of the kidneys, ureters, and the urinary bladder and the urethra. And its main function is production, storage, and expulsion of urine. Now the kidneys. Kidneys have several functions. One of the functions is excretion. It usually eliminates water-soluble metabolic waste and foreign substances as urine. Some of the metabolic waste include urea and creatinine. We also have regulation and here or homeostasis. It has homeostatic function in that it maintains appropriate fluid level and concentration of various electrolytes in the body fluids, and also maintains the normal blood pressure. It is involved in the mechanism which is usually required in blood pressure regulation, known as the renin, angiotensin, and aldosterone system. Renin is produced in the kidneys. It also maintains the pH of blood. Also, it has endocrine functions in that it produces renin, which is an enzyme which converts angiotensinogen into angiotensin 1. You can find other books which refer renin as angiotensinogenase, since it cleaves angiotensinogen into angiotensin 1, so angiotensinogenase. We also have, it also produces erythropoietin. Erythropoietin is a very important hormone. It regulates the production of red blood cells. And when you do cardiophysiology, blood physiology, you see that blood pressure, to calculate blood pressure, you take cardiac output multiplied by the total peripheral resistance. Now, this resistance is usually influenced by the viscosity, and the viscosity refers to the hematocrit. So if you have more red blood cells in the blood, the resistance will increase, and so will the blood pressure. It is also involved in the production of the active form of vitamin D, the 1,25-dihydroxycholecalciferol. Remember, in the kidney, 25-hydroxycholecalciferol is hydroxylated at position number 1 to give you 1,25-dihydroxycholecalciferol, which is the active form of vitamin D. So those are the functions. Now, this is the overall structure of the kidney. It has a cortex and a medulla. Okay, this is the kidney proper, or the renal parenchyma. Cortex, the darker outer, medulla, the lighter inner. Then we have a hilum here. This is the medial side, this is lateral, and then there are structures which enter and exit the kidney at the hilum. In the human kidney, the arrangement of this structure from anterior to posterior is renal vein, then renal artery, then renal pelvis, then we have the subsidiary branch of renal artery. Now, let's look at the renal cortex. In the renal cortex, you will find renal corpuscles. These spherical structures like that one, that one, that one, that one. Those are your renal corpuscles, okay? And they usually form ultrafiltrate from blood. Then we have cortical labyrinth. Cortical labyrinth refers to the area of the cortex that is found between these renal corpuscles, and the medullary rays. These are the medullary rays. Now this region here, that the one between that, those are your cortical labyrinths, and they have the proximal and distal convoluted tubules. Then medullary rays, there are projection of tubules between the cortex. Here is the cortex and here is the medulla. So there are projection of tubules between the cortex and the medulla, and they usually contain the straight tubules. Straight tubules refers to the proximal and distal straight tubules, and also the collecting ducts. Then one medullary ray and the surrounding cortical tissue. So one medullary ray and the surrounding cortical tissue form a renal lobule, okay? Renal lobule, not a renal lobe. a renal lobule. Please understand that. Then we have uriniferous tubules. Now, the functional unit of each kidney is the microscopic uriniferous tubule, or you might find some text saying that the basic functional unit, the basic structural and functional unit of the kidney are the nephrons. Okay, because uriniferous tubules refers to a nephron and its collecting duct. Okay, so nephrons usually produce urine, and the excretory collecting ducts conduct the urine from the kidney. Millions of nephrons are usually present in each kidney cortex. The nephron in turn is subdivided into two components. Each nephron has a renal corpuscle and a renal tubule. And there are usually two types of nephrons based on their location in the kidney and the organization of the peritubular capillaries. So these two mechanisms will help you to state which type of nephron it is. So you either use their location in the kidney or the organization of the peritubular capillaries. And using those two, we can either have cortical nephrons or juxtamedullary nephrons. Cortical nephrons, as the name suggests, they are found in the upper cortex of the kidney, and you'll find some text which refer to those nephrons as the superficial nephrons or the cortical nephrons. Then we have juxtamedullary nephrons, which are usually located near the junction of the cortex and the medulla. So they are located near the juxtamedullary junction. So they are juxtamedullary. They are located near the corticomedullary junction. So they are the juxtamedullary nephrons. Juxta means next, if I can remember. Yeah. Although all nephrons participate in urine formation, juxtamedullary nephrons usually produce a hypertonic environment in the interstitium of the kidney medulla that produces concentrated urine. So juxtamedullary nephrons produce more concentrated urine than cortical nephrons. We will see why. Here we are. These are your cortical nephrons. Yeah, these are your juxtamedullary nephrons. Okay, so they are distinguished by the structure and the function of the nephron groups and the organization of the peritubular capillaries. Cortical nephrons, they form the majority, 80%, and they are situated in the outer renal cortex, or they are found near the periphery of the renal cortex. They have a very short nephron. You can see very short nephron loop. The loop of Henry is very short and it usually deep. It dips into the superficial part of renal medulla or never leaves the cortex. So it can either dip only in the superficial part of the renal medulla, this is the renal medulla, or never leave the cortex. Their peritubular capillaries supply blood to their nephron loops. These are the peritubular capillaries. Then you have the juxtamedullary. Their corpuscles, they sit near the corticomedullary junction. You can see this is the corticomedullary junction here. They are sitting very near to the corticomedullary junction. They have a very long, you can see the loop of Henry here is very long, and it will burrow deeply into the renal medulla. And the loop of Henry is surrounded by vasa recta, and right here it is surrounded by the peritubular capillaries. Here are vasa recta, which arise from the efferent arteriole. Now, renal stroke Marfigian corpuscles. What are renal corpuscles? Renal corpuscles, they usually consist of a tuft of capillaries called glomerulus, which are surrounded by a double layer of epithelial cells known as the glomerular stroke Bowman's capsule. Then this capsule has an inner and outer layer. The inner or visceral layer consists of specialized branching epithelial cells called podocytes. Now, these cells are adjacent to capillaries, and they are long, and they have long cytoplasmic processes which completely invest the glomerular capillaries. Remember, the glomerular capillaries are fenestrated type of capillaries. From these cytoplasmic processes arise numerous smaller foot processes or pedicles. You can see where the word podocytes came from, that interdigitate with pedicles from adjacent podocytes and form tight-fitting filtration sheets. So podocytes are very important in formation of the glomerular filtration barrier. Okay, we will look at the glomerular filtration barrier. Then a thin semi-permeable filtration sheet diaphragm spans each filtration sheet. Then we have the outer or parietal layer of glomerular capsule consists of simple squamous epithelium. The renal corpuscle is the initial segment of each nephron, remember that. And the entry point of the afferent, where the afferent arteriole usually enter the renal corpuscle, and where the efferent vessels exit at that point, that is the vascular pole. And on the opposite end of the vascular pole of the renal corpuscle is the urinary pole, where the filtrate produced by glomerulus leaves the renal corpuscle. So maybe I draw that quickly. If this is my renal corpuscle, and this is where the afferent and efferent arteriole are entering and leaving, this will be a vascular pole, where this other side where the glomerular space will communicate with the proximal convoluted tubule, this will be your urinary pole. Hope you have understood that. Okay, now you can see here we are. This is your renal corpuscle. It has the parietal layer and the visceral layer which invest the glomerular capillaries. Those cells are specialized and they are known as podocytes. This is the vascular pole here. This is the urinary pole. Then you have a gap between the parietal and visceral layer known as the urinary space or the capsular space. Okay. Then here we are the same. This is the vascular pole. This is the urinary pole. Then you can see here glomerular capillaries. Then these cells here with the large ovoid nucleus, those are your podocytes. And yeah, let's move on. Then we have the mesangium. Now, these glomerular capillaries, they are usually supported by the mesangium. Now, the mesangium refers to mesangial cells, which are usually surrounded by the non-cellular mesangial matrix. So these are your mesangial cells, and you can see they are surrounded by the non-cellular mesangial matrix. Now, the mesangium usually form a mesentery-like fold over the capillary loop. Now, remember that the mesentery you had seen in the, if you have done the abdominal region, remember the small intestines, they are attached to the posterior abdominal wall by a mesentery. Such mesentery is the one which supports these capillaries. Now, the mesangium usually intervene between capillaries of glomerular segments. Mesangial cells contain, what is the function of these mesangial cells? They contain fibers which are similar, they contain filaments similar to myosin, and these mesangial cells contain angiotensin II receptors. And it is believed that stimulation by angiotensin causes those myosin-like filaments to contract, and by contracting, they will help to control blood flow through the glomerulus. Okay. Also, they are phagocytotic cells, and they also help in the maintenance of the glomerular basement membrane. The mesangium becomes prominent in a disease known as glomerulonephritis, which is also known as the Bright's disease, and they are of two types, primary and secondary. But that is story of another day. Here you can see the glomerular filtration barrier is formed by the endothelial cells, then the basement membrane, and the podocytes. Now, renal tubule. Now, it is divisible into several parts. We start with the proximal convoluted tubule, then the loop of Henry, then the distal convoluted tubule, then the collecting tubule. Note that along the entire length of the renal tubule, we have a single layer of epithelial cells. So it is simple, a simple epithelium all through, not stratified, simple only. So let's start with the proximal convoluted tubule. It usually begins at the urinary pole of the renal corpuscle. They have a tortuous or convoluted course lined by simple cuboidal to columnar epithelium with irregular or often sterate-shaped lumen. Also, they stain darker than distal convoluted tubule and ducts using eosin stain, so they are eosinophilic. So this, you can see this has a stated shaped lumen, also this, also this, also this. So these are the proximal convoluted tubule. Can you compare the staining property of this and this or this? You can see these are darker staining than this. So these are your distal convoluted tubules. Also, you can see the lumen here is stated shaped. The brush border here is very distinct, but here, here it is, you can see also the differences in the lumen. Here it is not stated. It is somewhat rounded. Now let's look at the weak descending limb of loop of Henle, or the proximal straight tubule. It is similar in morphology to the proximal convoluted tubule. So they are darker staining, and they will have a stated shaped lumen, and the brush border will be very distinct. This is a longitudinal section. You can see them very clearly here. Here it is, also this one. Okay, also this one. These are the proximal straight tubule in cross section here, here, also this one. But if you compare this one, the staining property of this one and this one, you can see these are darker staining. So this must be your distal straight tubule. Okay, these are your proximal straight tubule. Then let's go to the next part, which is the weak descending and ascending segments of the loop of Henle. And they are usually, it is a continuation of the proximal straight tubule within the medulla that makes a hairpin turn and returns to the cortex, lined by simple squamous epithelium. Note that this one, this one, this one, then also this one. Those are your thin descending and ascending segments of the loop of Henle. Then we have your distal straight tubule. It is also known as the weak ascending limb of loop of Henle. Its morphology is similar to that of the distal convoluted tubule. And here it is. You can see, if you compare this, these are your proximal straight tubule, and this, you can ascertain that these are darker staining than this. Okay, so these are your distal straight tubule. Also this one, I think also this in cross section, also this in the longitudinal. Okay, so this one, but these are your proximal straight tubules. Now let's look at the juxtaglomerular apparatus before we go to the distal convoluted tubule. Now, at the junction between the distal straight tubule and the distal convoluted tubule, that portion of the renal tubule usually come very close to the vascular pole. That portion of the renal tubule usually lie very close to the vascular pole of the renal corpuscle, and cells there are specialized. Now, how are they specialized? The cells, the cell walls, the cells of the distal convoluted tubule here, they are rather columnar. They are usually columnar, not cuboidal. And those cells are known as the macula densa. They are modified, the modified cells of the distal convoluted tubule, and they usually monitor the ionic constitution of the fluid passing across them. So these are your macula densa. Then these cells, the muscle cells of the afferent arterial wall, they are modified in that they are usually they have a rounded nucleus, and those cells are known as the juxtaglomerular cells. Okay, so juxtaglomerular cell. Then in between the two, we have race cells which form a race-like network interconnecting the two. So these three cells, juxtaglomerular cells, macula densa, and race cells, they form the juxtaglomerular apparatus, which is very important in the regulation of blood pressure. So if I can draw that quickly. So this is your distal convoluted tubule. This is your afferent arteriole here. So you'll find these cells here, the muscle cells here, they are modified. And also these epithelial cells here are modified. So these of the distal convoluted tubule are your macula densa. And these are your juxtaglomerular cells. Then in between them, interconnecting them, are the races cells. Okay. So let's continue. Now, the distal convoluted tubule, which usually empty the collecting tubule, here it is usually seen in cross section in the cortex due to its tortuous course. It's similar in morphology to the distal straight tubule. This, and you'd find some text saying that they are less than the proximal convoluted tubule, which is true. Even some texts suggest that the ratio of proximal convoluted tubule to distal convoluted tubule is 7:1. So the distal convoluted tubule are fewer. Here and here they are less staining. No, not less staining, right staining compared to this one, which are darker staining with eosine. Now let's look at the collecting ducts. They usually convey urine from the nephros to the to the collecting ducts within medullary rays. So they usually remember the medullary rays are composed of the collecting ducts and the straight tubules. Now, collecting ducts drain into a minor calyx at the apex of the renal pyramid, which is at the renal papilla, that is, and they're usually lined by simple cuboidal to columnar epithelium with well-defined boundaries between the cells. So you can see here clearly, you can demarcate the boundaries between these cells. And like here in the, like this is the proximal straight tubule, where you cannot demarcate the boundaries clearly, but here you can. Then we also have some cell types here. We have clear cells and the dark cells. So clear cells, their cytoplasm is clear like this one here. And dark cells, or the intercalated cells, they have their cytoplasm is somewhat darker staining. So the cell types of the collecting ducts are the clear and dark cells. So the dark cells are also known as the intercalated cells. Now, that was the kidney. Now let's go to the ureter. Now, ureters, they are muscular tubes that conduct urine from the renal pelvis to the urinary bladder. The wall of the ureter has 3 layers, namely the mucosa or the mucous membrane. Then we have muscularis externa, then the adventitia. So here you can see the mucosa refers to this epithelium and the lamina propria. And then we have a muscularis externa and the adventitia here. Here you can see the lumen is star-shaped because the mucosa has several longitudinal folds. Several longitudinal folds cause this lumen to be star-shaped, but when distended, when the ureter is distended, these folds disappear. Okay, mucosa, epithelium, and lamina propria. Remember, the epithelium is transitional epithelium, also known as the urothelium. And these cells, the surface cells of the urothelium, are the umbellar cells. One characteristic of the umbellar cells is that they are binucleate. You can see this, like this one here is binucleate, has 2 nucleus, and they have ability to change shape. So when the bladder or the ureter is relaxed, they are dome-shaped, but when it is stretched, they become flattened. You will see that in a few. Then you have the muscularis externa. It has an inner longitudinal layer and an outer circular layer. Please don't confuse this arrangement with that of the gut. Remember the GIT, the arrangement is inner circular, outer longitudinal, but this one is the opposite, inner longitudinal, outer circular, and that is for the upper 1/3 of the ureter. But as you near the bladder, an additional longitudinal layer is usually present, and in the lower parts of the ureter, it becomes inner longitudinal, middle circular, and outer longitudinal. Okay, so an outer longitudinal layer is added. That's one nearest the bladder. So adventitia is the outer fibrous coating consisting of loose connective tissue, and it contains numerous blood vessels, nerves, and lymphatics, and some fat cells, the adipocytes. Here you can clearly see the adipocytes. This is a blood vessel. Maybe this is an artery, and this is a vein here which has collapsed. Okay. Then let's look at the urinary bladder. And urinary bladder, its histology is similar to that of the lower parts of the urethra, ureter. So you can see here, it is a muscular bag where urine is stored temporarily and it's discharged periodically via urethra during urination or mituration. Then the wall of the urinary bladder consists of 3 layers: mucosa, muscularis externa, serosa, or adventitia. Now, the mucosa is lined by transition epithelium with no muscularis mucosa. Muscularis mucosa is only found in the GIT. Now, in an empty bladder, the mucous membrane is drawn into numerous folds that disappear when the bladder is distended. Some mucous glands might be present, especially near the internal urethral orifice. When the bladder is distended, or when it is filled with urine, the lining epithelium becomes thinner. This results from the ability of the epithelial cells of the umbrella cells to change shape and shift over one another. Here you can see here the bladder is relaxed. You can see they are dome-shaped. Okay, but here you can see it is filled with urine and becomes stretched. And you can see like this umbrella cell here, it is binucleate, and they have ability to change shape. Now, muscularis externa. The muscle layer in the bladder is very thick, and it consists of 3 layers: inner longitudinal, middle circular. You'll find some text saying middle oblique, but middle circular will do. Then outer longitudinal. Then what is the function of this muscle layer? It is responsible for the emptying of the bladder, and that is why it is also known as the detrusor muscle. So the detrusor muscle refers to this muscularis externa of the bladder. Then we have the serosa stroke adventitia. Serosa is not the same as adventitia, but the superior surface of the bladder is covered by the mesothelium of peritoneum, forming a serous layer. So serosa is found on the superior surface of the bladder, where the inferior part of the bladder is covered by adventitia, made of fibroelastic connective tissue carrying blood vessels, nerves, lymphatics, and also has adipose. So thank you for your attention, and please subscribe for more anatomy and biochemistry content. Thank you.